The title of my project is The Intergenerational Effects of Maternal Obesity on Offspring Mitochondrial Reactive Oxygen Species Production and DNA Damage. The Developmental Origins of Health and Disease states that certain environmental exposures during critical windows of development may have consequences for an individual's long-term health. Specific to my work, research has shown that offspring from pregnancies complicated by maternal obesity have a fourfold greater risk for developing childhood obesity and symptoms of metabolic syndrome. This hypothesis may serve an explanation for a portion of the continual increase in obesity rates among children. The overall goal of this project is to identify cellular mechanisms that increase the risk of metabolic syndrome in children exposed to obesogenic environment during development. Using a previously established Japanese macaque non-human primate model, we have previously shown that lean offspring from obese dams show signs of metabolic disruptions in skeletal muscle, including increased insulin resistance and mitochondrial dysfunction. Dams were placed on either a control or Western-style diet for at least 2 years prior to and during pregnancy and lactation. All offspring were weaned at 8 months of age to new group housing and fed a healthy controlled diet. Skeletal muscle samples from offspring were collected at 3 years of age. Our lab has shown a decrease in respiratory capacity and increased cellular signaling events associated with mitochondrial damage and recycling, also known as mitophagy, in skeletal muscle of offspring from dams fed a Western-style diet. Increased reactive oxygen species, also known as ROS, production can damage mitochondria and contributes to mitochondrial defects observed in obesity. Therefore, I hypothesize that maternal obesity increases offspring mitochondrial ROS production, leading to mitochondrial DNA damage without loss of mitochondrial DNA abundance. In order to measure relative mitochondrial abundance and ROS damage, we have developed a quantitative PCR-based method to measure mitochondrial DNA abundance and damage. 2 primer sets were designed. The first amplified a region that is about 150 base pairs apart as a measure of total mitochondrial DNA abundance. Amplification from a second primer set much further apart, about 10 kilobase pairs apart, will allow us to assess ROS-induced DNA damage. This is possible because mitochondrial DNA with ROS damage does not amplify as well, resulting in less PCR product. In our model of maternal obesity, dams placed on a Western-style diet had increased body weight and body fat in comparison to dams placed on a control diet as expected. Surprisingly, offspring body mass and body fat percent quantified at 3 years of age were not different by maternal group. Whole body insulin response was assessed in offspring by glucose tolerance test, also known as a GTT. Again, there was no difference in insulin production or the rate of glucose clearance from the blood. Taken together, the previously observed mitochondrial defects in skeletal muscle occur in offspring without clear differences in whole body insulin response or adiposity. To determine if differences in total mitochondrial abundance can explain previously observed differences in cellular respiration and mitophagy, we use the quantitative PCR-based method previously described to measure total mitochondrial DNA. An agarose gel of PCR product demonstrates specific PCR priming. PCR product was stained with pico green, and a standard curve of pico green fluorescence was used to quantify the amount of PCR product. Using this model, no differences were found in the amount of mitochondrial DNA PCR product, suggesting that offspring from obese dams have the same mitochondrial abundance as offspring from lean dams. More so, reduced mitochondrial abundance does not explain the differences in offspring muscle mitochondrial respiration. Moving forward, I will test for elevation in ROS-induced mitochondrial DNA damage to determine whether observed reductions in mitochondrial homeostasis are linked to elevated ROS production. If my hypothesis is true, I expect maternal obesity to lead to a persistent change in altering offspring cellular homeostasis, resulting in either reduced mitochondrial ROS production or suppressed ability to sequester ROS, and ultimately leading to mitochondrial DNA damage.